Hello, everybody. We are going to get started with our next speaker of the day, Alexandra, and we are going to have a short introduction from Rodrigo, who is unable to join us as he is in Portugal. So we'll start with Rodrigo and then on to our next speaker, Alexandra Kahn.
Hello, everybody. It is my pleasure to introduce Alexandra Kahn today. I apologize for not being able to do it in person. So Alexandra has been working on an important plant pathogen called Xylella fastidiosa. She has really spent a lot of time asking questions related to which plants does this pathogen colonize and how does it colonize these plants. And she's made some pretty interesting and important findings that I believe she will show to you today. I wanted to say a couple of things about Alexandra's work and her PhD very briefly. I will say that Alexandra went through all the steps of completing a PhD, from being excited to frustrated, seeing things work and seeing things not work. And in many ways, I believe that a major sort of event was when she was a little bit frustrated with some of her work early on and not really sure why it mattered. And she ended up going and presenting her work at an international conference. And she saw that everybody was really excited about the relevance of her research and why, she was able to sort of appreciate why the work was so important and impactful. And after that, it's fair to say that her work sort of took off, and I think it became clearer to her why, why it mattered. She has found that learning about the host range and specificity of this pathogen, essentially which plants it infects, it's a lot more complicated. And she's also done some work clearly demonstrating that the taxonomy of it, which is really important for quarantine purposes, it's far from clear. She has also done some work more recently trying to understand the origins of a disease in grapevines in California. And she led a very large field project that essentially showing that even for a disease that we've known and studied for about 100 years in California, that we still don't really understand the basics of symptom development and plant colonization. And much of that is because there's very little field work with infected plants. Altogether, I would say that Alexandra has explored a few different topics, and she has done an excellent job addressing very important questions in the field in ways that are very clear, even if at some point is essentially to tell people that we don't understand host specificity in particular, and that's really important to continue pursuing these lines of research. On a more personal note, Alexandra has been an amazing member of our group. She has been very sort of helpful. She has helped everybody and contributed to a number of different projects. She also has a lot of energy and positive attitude. At the same time, she's more than happy to, in my case in particular, let me know when she disagrees with something or things are not quite right, which is something that I really appreciate, that we have a good relationship, and she's very comfortable telling me when she disagrees about stuff. So she's also monitored lots of undergraduate students and interns, and she has been a very significant advocate for them, often convincing me that I should do things that I either forgot to do or that I should be doing to better support our undergraduate students in particular. So with that, Alexandra, congratulations, and I'm very sorry I cannot be there, but it has been a pleasure working with you, and I, in fact, look forward to continuing to work with you for a couple more years. So congratulations, and everybody. I hope you enjoy Alexandra's talk and learn a little bit about her accomplishments. So thank you.
All righty. Okay. How close should I stand? Good? Good. Cool. Well, as Rodrigo mentioned, I will be talking about both the associations between plant hosts and the pathogen Xylella fastidiosa, and also my various frustrations. So it'll be a nice balance. Yeah, thanks for having me. I'm really excited to do my finishing talk with so many smiling friends and faces around. All right, so kind of skipping the intro generally, so I can talk about a lot of my research, but these are some various plant diseases, all caused by the same species of bacterial pathogen. Briefly, you can tell that the plants look different because they're very different, like very distantly related plants from each other, but also maybe you can kind of start to see that the symptoms look pretty different. Up here we have coffee, which kind of just curls a little bit, but generally is kind of okay, whereas we have just like a dead grapevine and some dead olive trees. So just like the symptoms look different on the plants, the bacterial pathogens actually can exhibit pretty different phenotypes within the plants as well. So these are some images of plant xylem vessels, which is where this bacteria resides within the plant. And so this is from the left to right, almonds, coffee, and citrus. And you can kind of see these rod-shaped bacterial cells, and they look kind of different. So these like clumps and lines around the cells, these are biofilms. And so the formation of the biofilms, and also kind of the opposite, the biofilms make them stay in place, but also how they move throughout the plants is really important for host specificity and symptom development in these huge number of different plant hosts that this species of pathogens can infect. So in terms of my dissertation, it's going to be entirely about the relationship between these different hosts and the varied diversity of bacterial pathogens that infect the hosts. So I'm going to start out with the broadest chapter, talking about how the bacterial pathogens vary in their ability to infect, just like many different genera of plant hosts. And then I'll focus in on one introduction event and just talk about two of the important hosts, coffee and grape, forgetting what that is, a grape. And then finally, I'm going to just like zoom in on grape alone. And grape is the most important host here in Northern California, where we have lots of grape growers and also lots of Xylella. So for this first chapter, the main question that I was interested in answering is whether we could use the bacterial genome to try and understand relationships between plant hosts and Xylella fastidiosa strains. So these are some of these varied hosts: coffee, citrus, olive, sunflower, grape, more coffee, almond, blueberry, just like really all over the spectrum. I'm not a plant biologist explicitly, but I know there are some here, and they can probably tell you that these plants are not like in one group. They're not closely related to each other. They're really across the spectrum, but these different strains of Xylella have found ways to infect hundreds of different plants. Okay, so the first part of this project was just to try to understand, based on whole genome data, how these different strains that we had from different plant hosts from all over the world related to each other historically. It's actually not as blurry as I thought. Cool. So on the left, these are just a comparison of different phylogenies using different methods and different really subsets of these whole genome data. And so the kind of general overview is that using these different parts of the genome don't really recreate identical results. And this is really important because, as Rodrigo mentioned, there's a lot of like quarantine and policy issues surrounding these pathogens and the global plant trade. And people like to use different metrics to understand like how to classify a particular strain. So from this work, the first point was that it really matters which method you use to classify where your strain fits. And then once I had those, once I had a better understanding of how they related to each other, my next goal was to try to use that to understand the history of host specificity in these different strains. So over here, we just have another phylogeny. So it's just like a big family tree, and all the tips represent individual strains that were sequenced of Xylella. And on the outside, you can see what hosts they infect, just like in my little doodles. So up here, we have our grape population that I was just talking about, California. On the bottom, we have olive, and then just lots of other hosts. But one of the important results from this study, where I tried to infer what the ancestral hosts would have been at these various nodes, was this one that I've highlighted here. So this node represents the outbreak from Central America into Northern California about 200 years ago. And so with these data, we predicted with very high likelihood that the ancestors of this California outbreak, which has caused, economic devastation, huge loss in grape yield across California and more recently in other countries. So the origin of the strain would have been able to infect coffee. And so coffee isn't native to Central America, but it is grown quite heavily and has been for a long time and is really economically vital to a lot of Central American countries. So we have this evidence supporting other people who have also hypothesized that it's likely that the plant trade between Central and North America in the 1800s might have been responsible for this bacteria making it from Central into North America. And I guess one little side note is that coffee is also hypothesized to have been responsible for several other global outbreaks due to the trade. Everyone loves their ornamental coffee plants. All right, so I just had a slide of data that was put together, but I also just want to express that even though one of the things I learned throughout my PhD, and I've learned lots, is how to put together a decent looking slide, that it was so much more tumultuous than just that. So it's published now, which is really cool. This is my first author publication. But I did it during COVID. The first time I submitted, it was rejected. It also took like years of just figuring out how to manage the data and work with it just to get like a couple of tiny little figures. And then, worst of all, that conference that Rodrigo mentioned, this is the cover photo they used of me, going like this. So all of the trials and tribulations of putting together a dissertation. Okay, so then the second chapter is when I zoom in on just this one introduction event that I was talking about. So I wanted to know how Xylella fastidiosa adapted to novel hosts in California after this introduction event. So as I mentioned before, we have this hypothesis that it was introduced from coffee trade and maybe the 1800s, but some of the work we're doing now might push that date even earlier. And so in Central America, specifically, we've done a lot of work in Costa Rica because they have really great infrastructure in terms of their Xylella research. There's tons of Xylella in the coffee. Like some of the coffee farms have up to like 95, 100% plants that test positive for Xylella. However, the symptoms are extremely mild. Whereas in California, a lot of these great plants that get infected with Xylella are just like completely blasted. They just dry up and die really fast. So some of my questions really surround that dynamic. So the two main groups of questions. The first one I attacked via greenhouse work. So I wanted to know these California strains that have been adapted or at least existing in grape for the last several hundred years. I wanted to know, are they still able to infect coffee? And then if yes, are the strains highly virulent in coffee? So this question kind of goes back to here, like they're not really very virulent here, but they're very virulent here. So the question is, was there an overall increase in virulence, or was there just a shift to a new host, and in that new host, they're virulent? And then the second part was computational. So using more genomic data to understand if there's evidence of genetic adaptation to grape in these strains. And I was able to do this greenhouse work by getting a bunch of plants donated from pretty much the only coffee grower in California. So if you want some like very fancy coffee, ask me where. All right, so the greenhouse data. So after about 5 months in the greenhouse, about 75% of the coffee plants that we infected were positive with Xylella. The bacteria was thriving, but those numbers really plummeted. So after a year, about 5% of the plants were positive. And I just want to say that for some of these numbers for plant pathogen infections, sometimes you really need pretty low percentages to actually like have a bad infection in the environment. So after a year, 5% infected is not zero. It doesn't mean that they can't persistently infect. It just means that they can't persistently infect well. So, but generally, they're not really great at infecting coffee. And then the second question was, are they hypervirulent in coffee? So these grape strains, are they suddenly like really nasty, even in their old hosts? And so one of the metrics that we used was to measure the heights of the plants. So in red, on the left, you can see the control. So this is just inoculated with buffer, whereas the other three were inoculated with strains of Xylella from California. And there actually was some stunting that was significant in the beginning of the study. By the end, they were all kind of evened out with, you know, that only 5% positive. Maybe that has something to do with it. Maybe not. I'm still finalizing this section. But so they were vaguely symptomatic, but they definitely were not hypervirulent. It was not anything like the amount of virulence that we see in grape in California. So then the second part, so they do seem to be changing. They're not the same as, they're not able to infect coffee well, and they are able to infect grape. So these, yeah. So then what genes are associated with that adaptation to grape is the next question. I'm not going to go too deep into the computational methods, but the three general tools that I used were looking at gene gain and loss. So unlike probably most of the systems that you study, the bacterial pathogens can actually like lose whole genes and gain them very frequently through horizontal gene transfer. And then so we also looked for single nucleotide polymorphisms and looked for signs of positive selection. And so this led us to having like a pool of candidate genes that I'm still sorting through that at some point can be tested. But there are quite a few genes that show up as having an association with the grape strains. And some of them have pretty interesting functions. Some of them have to do with cold survival, which makes sense in terms of, you know, having an environmental shift alongside the host shift. One of them, PAR E, is actually a pretty cool one. It's a plasmid-associated toxin, and what it does is if a bacterial cell replicates and the next generation doesn't have a particular plasmid, it'll just kill it. So these are things that definitely could be associated with adaptation to a new host, but a whole nother set of things would need to happen to be certain of such things. But there are some things also that have to do with movement and biofilm formation, like I talked about in that first SEM slide. So there are some candidates that make sense, and it does seem like we do have some sort of shift in the bacterial genetics corresponding with this introduction event from Central America to North America. So finally, I dove deep into grape, specifically in Northern California. And I say finally, but this project was one that has lasted through my entire 6 years of my PhD. It started when I first showed up and they said, okay, we have this money, come, let's go to the field. But it's a really cool project that involved a huge, amazing crew of people, both in cooperative extension and other UCs, as well as grape growers in Northern California. And just one little issues slide before I get started is this email that I got after my first year of data collection. I'll just read one little bit. This is from the cooperative extension specialist, Monica Cooper, we worked with. She says, in bad news, they removed and replanted most of our infected Chardonnay vines, only 4 out of 15 remaining. Ugh, they can't seem to remember that we have a trial there. So that was a fun email to get. And then this is a picture of my mom who I went to visit. And this was about 2 minutes before I got a call from Rodrigo telling me that the -80 freezer crashed and all my samples were gone. So that was another fun event. But luckily, the 2-pronged approach to this project was fine with just 1 prong. The questions that I'll talk about at least today are first, what is symptom development like in Napa Valley? And how fast do the bacteria move through the plants? And anything else you want to talk about, find me later. Okay, so for this project, we inoculated many plants in Napa Valley with 1 strain of Xylella fastidiosa that's also from Napa. So some of the symptoms that we were looking for specifically are here, this is called leaf scorch, and it looks different in different grape cultivars, but you often see this like marginal reddening. You also have stunted plants. This one's pretty intuitive. It's a tiny plant amongst big plants. And that's all from one little set of bacteria. We also looked for shriveled clusters of grapes, which is another indicative symptom. And so all three of these maybe don't look so bad, but I just want to remind you all together, when they team up, this is what they can do to a plant. All right, so for our first question, we wanted to understand just what symptom progression looked like. And so this is a chart from the first year of the study. This is the inoculation year. So we inoculated the plants in April of 2017. And you can see in the first year across all three of the grape varieties that we used, we see these high magnitude peaks of shriveled clusters in the first year. And then later in the season, we see in orange here, a fair amount of leaf scorch. So while this might seem totally random and not like it matters at all, leaf scorch is always thought of as the first symptom that people in the field use to detect Xylella infections. And it's thought to be the most prominent, and it's what people use to actually try to stop Xylella from spreading. There isn't really a treatment for Xylella, so the only thing you can do is take down the plant before it can spread to new plants. So it's really important to have some information about what the very first symptom is so that we can identify it as fast as possible and prevent spread. And then, so in the second 2 years, we actually see pretty different symptoms. We see a lot of stunting in the early seasons across all three varieties. And then again, later in the season, we see this leaf scorch. So not as much of the shriveled clusters, but they are still present. So yeah, pretty different from like the point of inoculation and that year as opposed to subsequent years. And the other question we were interested in is understanding how the bacteria and also the symptoms that are associated with the bacteria move through the plant and how fast they're able to move through the plant. So it's a lot of data all in one slide, but I want to try to break it down. So each of these grape plants are kind of like shaped like a candelabra. It's actually nice for like visual analysis because they're kind of 2D, even though they're not. So what we did is we inoculated them all on like all the way on one side, on the western side of the plant. And then we tracked across the plant to see how fast it spread. So in the first year, the symptoms up here are only on the west and midwestern part of the plant. And on the other side, we don't see any symptoms at all. So in the year of inoculation, the symptoms are really like focused by the inoculation point. Whereas in years 2 and 3, they're very evenly distributed throughout the plant. We can talk about why more later. And the bacterial populations show exactly the same pattern, which is pretty cool. So in summation, I want to talk briefly about my process, which Rodrigo mentioned, I guess, was very cyclical. But for all of these projects and all the other ones that I've done that I'm not talking about, the beginnings are really always so hard. It takes years of like failing and trying to figure things out, things that get screwed up, freezer samples that get melted. But then all of a sudden, things start working and I start making progress. And this goes by so fast every time. It just sucks. And then eventually I have a pretty firm grasp on what I'm doing. And while you think that might be like the right stage to be in, every single time it feels like if I understand what I'm doing, it's too easy and my data is worthless, which I guess is why it helps, you know, present and hear other people say nice things to you. But yeah, over and over again. But, you know, after all of these cycles and putting together a PhD, I can actually look back and like see what I've done and feel proud of it. And it doesn't feel as daunting. So I'm just going to keep doing it. As Rodrigo said, I'm going to do a postdoc here in ESPM now. So yeah, just more of the same. Yeah, so I wanted to thank first Rodrigo and my lab group. Rodrigo has been just, you know, amazing throughout the years. It's really, I feel really lucky to know that I've been able to have him have my back this whole time. And he's put together just such a great group of people over the years, time and again. I also want to thank so many of my fun ESPM friends and my amazing cohort for lots of good times over the last 6 years. And I want to thank my family, some of which I think are probably on YouTube right now from all parts of the country listening in. And just thank you so much for your perpetual support and love. And I'm going to plug this weekend my side hustle. If you want to support me, buy some bug themed mugs this weekend. Yeah. All right. So any questions? Very blurry.
Excellent talk. I also love when you talk about the fails. I feel like we can all relate. So thanks for sharing every part of the process. I was curious about because the grape strains are so much more virulent. I can never say that word.
It sounds fine. Go with it.
Virulent than
Perfect.
in grape than in coffee.
Mhm.
But then in your phylogenetic analysis, it looks like coffee is like spread to a lot of other things. Do you see the same trend in other plants that it's got when it's like become from coffee to those other plants? Is it also the same level of virulence as in grape? And is that kind of a similar process?
Yeah, actually, there's another really, really bad outbreak right now in Italy. Let's just pull up a picture. Find it. It's all the way back. So this in the bottom right is in Puglia, so in the heel of Italy. And so this is another outbreak that's been hypothesized to have been spread via coffee. And it's really, really devastating. And they like talk about like the strain, they call it ST53, and they're like, this is the most virulent strain ever. But in some ways, like it is a really similar situation where in coffee, the strain doesn't really do very much. And then in the other hosts in Italy, it's just been like awful. So that's part of the reason why I wanted to focus on coffee. It's just like time and again, it keeps popping up as really important in terms of like global plant introductions. And I think part of the reason is that maybe it has like a low percentage that get infected and that's enough. Or maybe it has persistent infections that just don't cause a lot of symptoms. And because of that, it's able to bypass like security checks. Because even when I did like do this testing and found like the 5% positive, in the tests that I did previously, so it was like 8 months, 12 months, there are a lot of plants that tested negative and then positive 4 months later. So I think that like there's something going on with coffee and its ability to like not really be symptomatic and maybe have spotty detection. And that's part of and aside from it being so cute, so everyone wants it in their home, it's part of the reason maybe why it's been such an issue. Yeah.
Yeah, thank you very much. Did the vineyard owners or managers have any resistance to the inoculations?
So a lot of our funding comes from this really great organization that's all focused on Pierce's disease and its vector in Southern California, the glassy winged sharpshooter. And so this project was started partly just because after Pierce's disease became such an issue time and again, the cooperative extension specialist at various conference was just like getting people who wanted to be involved. So this wouldn't have happened if the growers weren't interested in getting more information about the progression. But even so, as I mentioned, it was hard. Like even the growers who were excited about it kind of started freaking out when they started to see symptoms. And so they got rid of a bunch of the plants. So it's not to say that we were sneaking them in or anything. It's just that, you know, when your livelihood's on the line, it's hard to commit to having a research block that is certainly going to negatively impact that part of your vineyard. So it's complicated, but I mean, as someone mentioned before, or David, I think in the morning, like the cooperative extension network in California is really so fantastic. And because of those amazing connections that the specialists have with the growers and how much trust there is there, that's the only reason we're able to do projects like this at all.
Thanks so much, Alex. This was a wonderful talk and so fun to see what you've been up to for the past 6 years.
Right next door.
Right next door. I had no idea. I'm wondering, is there any benefit to the bacteria to being more sort of devastating to the host? Like, is there a reason to think that the bacteria would be actively moving towards a more virulent place? Or is that just sort of like an unfortunate byproduct and sad for grapes?
Not necessarily. I mean, so there have been some studies that show, so I didn't talk about this at all, but Xylella is insect vectored. So it's not an environmental pathogen. It can't spread through contact or through the water. It can only spread by xylem feeding insects that will suck the sap and then spit it out into a new plant. And so insects don't prefer symptomatic plants. If they have a choice, they'll choose plants that look healthy. So there's not necessarily like a benefit in that way for like increasing the symptoms and becoming more virulent, because you're less likely to be passed to the next generation and another plant. But this is also like, I also didn't talk about this at all. This is an agricultural system. Like this is very different from a natural environment. And Xylella is present in many natural environments and typically doesn't cause really intense disease in the same way that it does in urban or agricultural systems. And I think a big part of that is just like having monoculture and the ability to just like, if grape came here 200 years ago and every generation it just needs to survive in the same type of host plant as opposed to going in a forest from plant to plant to plant, it's just going to intensify its behavior and ability to infect this one plant. So that certainly has to do with the way that it's adapting and causing sickness in plants.
Great talk. Right here.
Hey.
Yeah. So one, I just wanted to validate the whole like failing thing. I think that's like a fantastic thing to show because it's just true from all our experiences. And yeah, I also, whenever I get a positive result, I'm like, I did something wrong. Like there's no way that's right. Like I'm so skeptical. And I'm so glad you mentioned that as well. Would love to hear you talk more about that. But my question is Xylella sort of like, is it friendly to like genetic engineering techniques? So if you wanted to say, do some gene knockdown experiments or knockout for those candidates you were thinking about? Is that something you could do, or do they not grow in culture? Or,
Okay,
yeah.
so it's culturable, which
Let's
is
go.
really great because a lot of plant bacterial pathogens are not culturable at all. It grows really slowly, and it's kind of a pain in the butt. So there have been some work done, especially there's one lab at Auburn that does some really great like knockout work. We've tried to do some here, and I feel like, we're all kind of dabblers. So maybe we just like are too chaotic to get it right, because most of the stuff we've tried to do here hasn't been that effective, but it's definitely possible. It's just not, it's not as easy as it is in some systems.
Totally. Thank you.
About your coffee plant. I have one.
Oh yeah, I only know how to kill plants. People always are like, how do I keep my plant alive? And I'm like,
No, I know. So my question is, if your coffee strains that you got here in California are different from the ones in Costa Rica.
mm-hmm. Yeah. So, in Costa Rica, this is like the native range of this pathogen. There's a huge amount of diversity of the different strains that infect coffee. We actually have a project right now trying to better understand the diversity of strains in Costa Rica. And it seems like there was just one introduction event several hundred years ago into California that is then diversified into like a whole different set of strains. So those are the ones that we were using for this project. But they have done some experiments at the University of Costa Rica in San Jose, trying to infect grapevines with the strains from coffee, and they haven't seen infections there, which really does imply that there is like a change in these strains in California that allow them to infect grape, whereas the ones in Costa Rica, at least, there are many strains, but the ones that they've tested haven't been able to infect grapes. Thank you.